Hi, and welcome to the VieSID Press-release. In this video, we report on articles selected from the dental literature published in October 2022 that we hope might be of interest to the VieSID community. So let's begin. In this AJO-DO paper, Kongson et al. Evaluated differences in post-surgical airway volume of subjects going through maxillomandibular advancement with or without genial tubercle advancement. They considered 26 patients with moderate to severe obstructive sleeping apnea and measured the airway volume before and after surgery. They found a significant increase in airway volume between the pre- and post-operative condition based on CPCT images, but did not find significant differences in the post-operative condition of patients undergoing maxilloromandibular advancement with or without genial tubercle advancement. Thus, the authors concluded that the procedure for genial tubercle advancement might not be helpful. I cannot discuss the clinical implications of this surgical procedure, but I just want to point out that according to the results reported by Kongson et al., no differences were found at the end of the treatment between the two groups, but the patients who underwent the surgical protocol with genial tubercle advancement were those with the lowest initial airway volumes. In fact, this might also be the reason why the maxillomandibular advancement with the adjunctive procedure was performed in these cases. Therefore, the initial condition was less favorable than that of the other patients, but managed to reach similar outcomes. The AJO-DO issued also a paper by Rozzi et al., who investigated the changes in curvos P obtained in two patients' cohorts, one consisting of 30 subjects treated using the Invisalign, and the other consisting of 32 subjects using a standard edgewise full fixed appliance. Both groups were treated for approximately 2 years. The leveling of the curvos P was measured as in the illustration based on the distance from each cusp to the plane formed by the points between the incisor and the distobuccal cusps on the M2S, so the second molars. Cephalometric parameters were also investigated. With both kinds of treatments, the authors observed changes in the curvos P. However, they reported that the leveling with the edgewise full fixed appliance was obtained via the extrusion of the posterior teeth and proclination of mandibular incisors, while changes using the Invisalign were achieved by the intrusion of the incisors. Although the authors stress the importance of the leveling of the curvos P as the primary treatment goal, we should assume that the curvos P is only one of the clinical parameters of relevance. So all relevant parameters should be considered while evaluating the validity of these orthodontic techniques, and possibly the effects of the treatment on the function of the masticatory system should be assessed too. The Annals of Anatomy invited Kyle et al. To review the dental research published in its special issue, Dentistry, in the last 10 years. This issue was founded with the aim to allow an interdisciplinary exchange of knowledge between medical professionals, dentists, and researchers. This review considered therefore 70 papers, of which 46 were experimental, 16 clinical, and 8 were reviews. The experimental articles included 16 animal studies, 12 human studies, 11 using theoretical models, and 7 in vitro studies. These works focused on oral surgery, prosthodontics, orthodontics, periodontology, dental medicine, radiology, biotechnology, and anatomy. Kyle et al. Reported on findings within the fields of implantology, bone regeneration, gingival regeneration, dental and maxillofacial anatomy, orthodontics, and aesthetic dentistry. Here, I report the summary of the anatomical findings relevant to dentistry. A, bone is formed under the influence of shear forces. The dental follicle transmits the forces from the tooth to the alveolar bone and serves as a source of periodontal tissues. This information was reported in Kylie et al. 2016. The lateral pterygoid muscles is involved in almost all movements of the temporomandibular joint. The two-headed variant is the most common, and palpation of the muscles, which is important for diagnostics, could be learned with the help of an MRI. This information was reported in Stelzel-Müller et al. 2016. In point C, they reported that CBCT should be the gold standard for evaluating maxillary sinus anatomical variation, as well as for assessment of the intraosseous organization of the anterior part of the human mandible. Accurate anatomical information may help to avoid surgical complications. This information was to be found in Lozano-Carrascal et al. And Pristanska et al. 2018-2020. In the Annals of Anatomy, Ma and colleagues reported on their analysis of the condylar and incisors movement in subjects with healthy temporomandibular joints eating French fries. In patients with unilateral TMJ derangement, it was observed that the affected glenoid fossa is deeper, with a steeper anterior slope. This asymmetry was found to be associated with unilateral mastication, with the mastication side mostly on the same side of the affected TMJ. Thus, the study was performed to understand the possible effects of habitual unilateral chewing side on the TMJ. For this purpose, the volunteers were instructed to chew unilaterally. The mandibular movements were recorded using a motion capture system, the NDI Polaris Viga. Eventually, the authors included 12 young adults, asymptomatic and without patent malocclusions or asymmetries. The trajectories for the incisor movements were collected based on the Frankfurt plane. They also calculated the trajectory of the condyle based on arbitrary axes passing between points anterior to the condyles, as in the figure to the left. Moreover, condylar acceleration during unilateral mastication was recorded. The incisal biting was rather symmetrical, while the chewing showed different condyle movements. The displacement for the non-masticatory condyle was significantly larger than that of the masticatory condyle. The average lateral displacement of both condyles were relatively small, around half of a millimeter or less, while the trajectory angle of the masticatory condyle was significantly greater than that of the non-masticatory condyle. The average duration of one single masticatory cycle was 0.76 seconds. Obviously, the acceleration of the masticatory condyle, which shows a shorter displacement, was higher than in the contralateral side. The working TMJ acts primarily as a fulcrum with an average displacement of the condyle of 0.18 millimeter, while the non-working TMJ acts as a gliding joint with a 0.41 millimeter displacements. Thus, Ma et al. Concluded that due to plasticity of the TMJ, the long-term unilateral mastication might lead to forward remodeling of the anterior surface of the articular fossa on the non-masticatory side, while deepening the articular fossa on the masticatory side. In the Archives of Oral Biology, we found a work by Calmari et al. Exploring the relationship between the polymorphism of the gene for collagen type 2 alpha 1 chain, abbreviated as COL2A1, and mandibular skeletal malocclusion. The authors postulated that the mandibular condylar cartilage can be involved in the development of mandibular retrognatism or prognatism. The condylar cartilage is divided into superficial, proliferative, chondrocytic, and hypertrophic zones. In the proliferative zones, proteins regulating chondrogenesis are expressed. After the formation of chondrocytes from the mesenchymal cells, collagen type 2 alpha 1 chain is expressed in the chondrocytic layer. Here you see what the collagen type 2 protein looks like. Collagen type 2 contributes to the formation of hypertrophic cells that eventually undergo apoptosis, and through endochondral ossification, bone replaces the cartilage. This process occurs already in the 13th week of embryogenic development. The allele expressing collagen type 2 can present single nucleotide polymorphism, which result in changes in the protein structure at a particular region of the protein. Here is color-coded. The authors use this single nucleotide polymorphism as genetic marker to detect possible association with dysgeusia. Thus, they considered 102 skeletal class 1 individuals, 81 class 3, and 82 class 2, sampled from the North Iran population of Mazandaran. They sequenced the gene and found the occurrence of the possible genotypes CC, cytosine, and CT, cytosine-thiamine, but did not find any TT, thiamine genotype. They also found that the T allele was associated with retrognatism, but not with prognatism. To explain the occurrence of retrognatism in case of CT genotype, we should consider that the protein resulting from the CT genotype substitutes the amino acid serine with glycine, disturbing the function of that domain of the protein, which I remind you allows for the ossification of chondrocytes. Thus, the CT genotype results in unfavorable ontogenetic development of the condyles and mandible. In the same journal, Young et al. Published a paper discussing the functional interpretation of the enamel microstructure in permanent premolars. In particular, they investigated the density and length of the Hunter-Schreger band configuration. Hunter-Schreger bands consist of an optical phenomenon generated by changes in direction between adjacent groups of enamel rods. In particular, there are elevated and areas and more depressed areas. And the longitudinal section under oblique reflected light, the elevated areas appear as light, and the depressed area are dark because of the shadow produced by the elevated areas. This optical phenomenon therefore originates from the fact that the groups of enamel rods are perpendicular to each other and are crossing each other, so that in a section we can find areas called diazones and others called parazone, depending on the orientation of the enamel rods. This decussated structure of the enamel lends the enamel resistance to fracture. So whenever a crack occurs, it propagates until it founds the resistance of the perpendicular oriented enamel rods. It is known that the functional cusps of the molar teeth, the upper lingual and lower buccal, possess thicker enamel than the guiding cusps. The authors wanted to test whether the functional cusps also possess a higher density of decussated enamel, namely higher density of Hunter-Schreger bands. They also wanted to investigate whether the relative length of the Hunter-Schreger bands differed. They considered 41 upper and lower teeth, including first, second, and third molars, which were already sectioned for a previous study. The sectioned surfaces were imaged at high resolution. As visible in the figure on the left side, the Hunter-Schreger band's density was calculated by dividing the number of bands by the DJ length at the cuspal and middle area of the buccal and lingual sides of the dental section. Additionally, the thickness of Hunter-Schreger bands relative to the total enamel thickness was calculated, as is illustrated in the image on the right side. The authors found out and concluded that the functional cusps display higher Hunter-Schreger band packing density and a relatively thicker decussed enamel than guiding cusps. As such, guiding cusps are intrinsically more susceptible to enamel chipping than functional cusps. The enamel of mandibular molars is generally less decussed than that of maxillary molars. As such, mandibular molars are likely more susceptible to enamel chipping than maxillary molars. These interpretations are consistent with the reported dental chipping patterns in both humans and non-human primates. The authors suggested that caution should be exercised in the interpretation of dental chipping frequencies, since other factors, in addition to decussation, contribute to enamel's biomechanical properties. You might also be interested in the Dental Clinic special issue on dental biomaterials. While I gladly share this information with you, I will not go into the details of this compilation of articles, which focus on dental adhesives, resin-based composite, dental light curing, bioactive restoration, dental material, digital technology, ceramics, and more. In the Journal of Prosthetic Dentistry, Thompson et al. Compared the mounting of maxillary casts using two arbitrary cast transfer system, the Coy's dentofacial analyzer and the Panamount phasebow, to the kinematic phasebow. Thus, they mounted the cast of 15 patients, and for each patient, made 3 repetitions. Comparison between the 3 systems was made by measuring the following parameters on pictures. The distance from the articulator axis to the incisor edge. The angle between the occlusal plane and the horizontal plane of the articulator. And the angle between the incisors plane measured at the canine tips and the horizontal plane of the articulator. The outcomes show that the incisor edge length on the occlusal plane means differ between the Coy's and the other phasebow systems, while the incisor plane did not. The study showed also that the means for the distance at the incisor edge and the occlusal plane angle differ significantly between the Coy's system and the other 2 systems. The analysis showed that there was a variance around the means for the kinematic phasebow and the paramount phasebow, which is to be expected considering the individual variation within the sample of 15 patients. Instead, the variance for the measurements obtained using the Coy system were very much lower. So despite the clinical statement of the authors, which claims that clinicians are justified in using the Coy's dentofacial analyzer, these results raise a red flag because indicate that the individual condition of the patient is not reflected in the articulated casts when mounting with the Coy's system. This might lead to problems when changing the vertical dimensions of occlusion. So we have come to the end of this presentation. I invite you to contact us in case you want to know more about our activities. Otherwise, be ready for next press release. Best wishes to all.